Zonal President, Central Zone, to provide and welcome and introduce the speaker.
Thank you, Dr. Prameshwari. Good evening to all, each and every one you. At the outset, I am welcoming each and every one of you for being here with us to listen our eminent speaker, Professor Nakkeeran. We all are very pleased to be able to welcome Professor Nakkeeran as well as all the distinguished participants. As such, Professor Nakkeeran has no need to introduce. But as a customary, I am introducing Professor Nakkeeran. He did his graduation from TNAU Coimbatore. He did his postdoc from University of Manitoba, Winnipeg, Canada. In his 26 years of services, he received many prestigious awards. These are Tamil Nadu Scientist Award from Honorable Chief Minister Tamil Nadu in 2017. He received Pesticide India Award, P.R. Verma Memorial Lecture Award, and R. Prasada Lecture Award from Indian Society of Mycology and Plant Pathology. Best Researcher Award, Best Extension Worker Award from TNAU Coimbatore. He is a fellow of NABS, Indian Society of Mycology and Plant Pathology, ISNS, many societies. He served as an editor-in-chief of Journal of Mycology and Plant Pathology and editor of NABS newsletter. He served as an EC member in these societies, ISMPP and NABS. Apart from this, he guided 6 PhD scholars and 10 MSc students. He published 154 research papers and 30 book chapters at national and international repute journals with the citation index 2501 and H index 22 with the RG score 31.6. He did tremendous work on the trichoderma viridi. In that way, he developed technology on mass production of trichoderma and popularized it through public-private partnership, PPP mode, between 120 stakeholders across the India. He organized trichoderma and PGPR training workshop at Nepal, and Tanzania, Bangladesh, Cambodia, Ethiopia through the financial support received from the IPM CRSP, Virginia, USA. He served as a consultant to the government of Bangladesh for establishing trichoderma production technology through the Swiss contact at Bangladesh. Not only Professor Nakkeeran did alone this, his student recently received a Jawaharlal Nehru Award of his thesis through ICR during 2020. So now I'm again inviting Professor Nakkeeran to deliver his talk on understanding the endophytes mediated induction of defense genes for the management of pathogen of global significance. Now, floor is yours, sir. Welcoming you again. Thank you, sir.
Thank you, madam, for the nice introduction. And so I think now the slides are clear. Am I correct? Can all of you, can you see the slides, Dr.
Yes.
Ritu?
Yes, sir.
Yes,
Yes,
sir.
visible.
Okay.
Visible.
Okay, thank you. At the outset, I would like to thank Indian Phytopathological Society for conducting this Platinum Jubilee Lecture Series 2021. So really, I am very grateful for the society. As a member of the society, that I have been given an opportunity to have an interaction with the learned scientists in the society. So in this juncture, I would like to present on understanding the endophyto-mediated antimicrobial action and induction of different genes for the management of pathogens of global significance. What's the lesson that the global market has given to us? So this is the one which we have learned now, where if you see the investment in microbial discovery and development is getting on increasing, where the microbial products is found to be growing at a very faster growth rate because it has an improved market access for biopesticide and biostimulant. And there is the integration of the cost-effective alternatives through integrated crop management strategies, which involves the application or the usage of these bioorganisms and biostimulants So this has resulted in contributing around 60% market share in coming years. So this is the global intelligence report, which is international survey report, which has been given by Dunham and Trimmer, where the major global share on biopesticide is mainly by the 4 to 5 major companies, namely Valent Bio, Copert, Certis USA, Bayer, and Syngenta. So these are the major players across the globe on biopesticide industry, where the microbial biopesticide, they play a key role in the multinational portfolio, because 100 products has been registered by Brazil, 125 products has been registered by European Union, and 420 products has been registered by United States of America. So if we see the registration of a chemical pesticide or a chemical fungicide from the research to development, it takes at least of 11.8 years, average 12 years it requires. And they had to invest around $286 million US for developing a particular product. So in these circumstances, during the era of organic cultivation, why can't we discover and deliver promising endophytes for the humanity? So in this case, so let us see what is an endophytome. So they are nothing but a complex microbial communities which colonize the interior portion of the plant pots in any part of their lifetime. So in this case, we should have an increased awareness on host-associated microbiota. So to understand the host-associated microbiota, how does the plant plan its carbon for budgeting towards the buildup of rhizomicrobiome and the endomicrobiome? So in this case, if you see, the diversion of carbon is around 20 to 30% of the carbon as a photosynthate in the leaf is diverted to the root region in terms of secreting the secondary metabolites like carbohydrate, amino acid for the root cap development, organic acid, mucilage, and vitamins. So these in turn decides the multiplication of the microbes which is present in the non-rhizosphere region to the rhizosphere region, and the rhizomicrobiome which is getting adapted to this condition. So they move into the endosphere based on the interaction between the rhizosphere and endosphere. So the organism from rhizo can move into the endosphere. So if you see what is the ports of endophytes immigration point of the entry, so they enter through the root cap, root hair, emerging roots. So after gaining the entry, they enter into exodermis, move into the cortex region, and to the endodermis and to the central cylinder. After gaining entry into this, it moves into the stem region and the colonization takes place in the vascular cambium, xylem, phloem, peridome, endophytes, and the xylem rays. So these are the regions where it goes and multiplies and colonizes the endosphere region. So though the colonization is there, the determinants of the endophyte abundance and its assembly is decided based upon what is the genotype of the crop, whether the developmental stage is found to be favoring the multiplication of these endophytes, and the soil type, geographical location, growing season, and the plant organ. So all these decides the abundance and assembly of the organism. So if you see the analysis of the data which is found to be available for us, 10 to the power 5 to 10 to the power 7 endophytes can be noticed per gram of the root tissue, 10 to the power 3 to 10 to the power 4 per gram of leaf and stem, whereas in the case of the flowers, fruits, and seeds, it's around 10 to the power 2 to 10 to the power 3. So this is the distribution pattern of the abundance of the microbes depending upon the different plant pots. And if you see the composition of the endophytes as a whole, the major share is by the proteobacteria, which shares around 50% of the population. 10% is by actinobacteria, firmicutes 10%, and bacteroids. It caused by 10%. The rest of the other occupies other 20%. So in this case, when these endophytes are having a major share in the colonization, so why can't we explore the endophytes for induction of immune response? So that based on our studies, I would like to share the information pertaining to the endophytes diversity for the management of viral pathogens, bacterial pathogens, and fungal pathogens. So in this case, let me move from banana to potato to that of the other solanaceous vegetables. So in the case of the endophytes, so the banana is one of the major crop. It's a globally important crop where the endophytes are found to be playing a major role. So if you see the genotypes which is found to be available in the nature, that is the blessing of the Almighty which has given us. So certain genotypes are found to be totally resistant for Fusarium azoospermum cubensi. So why this resistance mechanism? So just like that of the human gut microbiome project, so whether these microbiomes living as an endophytes in resistant genotype, can they play a role in resistance development in the plant system against the Fusarium wilt or not? So based on that, we have analyzed the diversity of the organisms, where these are the diversity where 6 different groups of organisms are found to be present in YKM5, which is a resistant genotype, and in the case of the Carprovoli, which is susceptible to that of the Fusarium azoospermum cubensi. So the population is found to be comparatively 50% lesser than that of the resistant genotype, and this is the diversity of the organisms which is found to be distributed in the root, culm region, pseudostem, petiole, leaf, bract, fruit, nectar, and flower. So this paper we have recently published in Environmental Microbiology by one of our student and our team we have published. And this is the diversity of the organisms which is found to be present in the susceptible genotype. So here, the data which we are analyzed, which clearly indicate that the culturable endophytome is found to be diverse in nature in the case of both resistant and susceptible genotype. So if we analyze this, the interaction, suppose the common microflora which has been shared between both the resistant and susceptible genotype here is, it is Rhizobium, Bacillus, Agrobacterium tumefaciens, Subtilis, Klebsiella, Bacillus amyloliquefaciens, Bacillus cereus, and likewise Pseudomonas mendocina, Pseudomonas gusicinae, Ochrobacterium intermedium. So these are the common microflora which is found to be associated in both the resistant and susceptible genotype. Whereas there was a 31 different types of bacterial endophytes were found to be highly unique in the case of YKM5, which was not found to be observed in the case of the susceptible genotype, where there they were having 30 different unique bacterial species, which was not found to be noticed in the case of the resistant genotype. So here we had a researchable issue that why can't we try these organisms which is found to be diverse in nature in YKM5 to assess its antifungal action and to compare that with that of the microbes that has been associated with that of the susceptible genotype. So this is the antifungal activity where we have done for all the 62 organisms in YKM5 and for the 31 from that of the susceptible genotypes, where here if you see irrespective of all these things, the Brachybacterium paraconglomeratum, Ochrobacterium intermedium, Pseudomonas aeruginosa, Ochrobacterium stritici, Brucella militensis, Achromobacter siloxidens, Myriades odorotimimis, Beijerium fleminensis, and Leclercia adenocarboxylata. So in all these cases, so they were having the capability to suppress the pathogen Fusarium azoospermum cubensi to the maximum of around from 20 to 70% inhibition of pathogen was found to be noticed. So this is the data where they represent how the pathogen was found to be suppressed even up to 70%, whereas 10 isolates were found to be having the suppression level less than 20% of the Fusarium infection, Fusarium mycelial growth was reduced by 10 different pathogens up to 20% level, whereas in rest of the cases, around 30 pathogens where 30 bacterial endophytes, they were able to suppress the inhibition of the mycelial growth of FOC to the maximum extent from 30 to that of 70% inhibition was found to be observed, where the maximum inhibition in the case of the bacteria which has been isolated from that of the susceptible genotype, where they were able to reduce the growth of the mycelium of the pathogen Fusarium azoospermum cubensi up to the level of 20% inhibition was found to be noticed in this particular case, except Enterobacter cloacae, which had 40% inhibition. But what we did is, whatever the organisms in resistant genotype which has the maximum inhibition of more than 50%, we selected those organisms. So this is the case where the pathogen has been suppressed. So why this suppression is there? So what are the metabolites that has been diffused into this region? So what are those metabolites that contribute for the suppression of the pathogen which is found to be associated in this particular wilt disease? So here we extracted these metabolites and test verified using the diffusion method, and they were also found to be playing a vital role in suppression of these organisms. So irrespective of all the isolate that we had, the effective isolate that we had tested, the Pseudomonas aeruginosa, Ochrobacterium intermedium, Brachybacterium paraconglomeratum, Ochrobacterium tritici, Brucella militensis, Lachesia adicarboxylata, Achromobacter xyloxidens, and Myriadus odorotimimus, and Beijeringia. So all these organisms, the metabolites extracted from the top, the zone of inhibition, was having the capability to suppress Fusarium oxysporum cubensi. So the network analysis that we did here based on the monotrophic interaction and ditrophic interaction, it reveals that the isolates Bacillus velicensis, when it interact with the top, the Fusarium oxysporum cubensi, it produced the metabolites like linoyelaidic acid, 5-hydroxymethyl furfural, in addition to that of clindamycin. Next to that, in the case of the Brachybacterium paraconglomeratum, during the ditrophic interaction with the Fusarium oxysporum cubensi, it resulted in the production of clindamycin, which is both shared by both Brachybacterium paraconglomeratum and Bacillus velicensis. In addition, it produced octane, acetamide, valproic acid, cytosterol, mucic acid, aminocaprylic acid, nonanol, and acetidine. In addition to that, Brucella militensis, this was also found to be associated during the interaction with that of the FOC. This was also producing linoyelaidic acid. So this particular linoyelaidic acid and the 5-hydroxymethyl furfural, mucic acid, aminocaprylic acid, nonanol, acetidine, clindamycin, octane. So these compounds were found to be shared commonly between the major organisms which are having the capability to suppress Fusarium oxysporum cubensi. So because of that, so these molecules, biomolecules which has been developed during the interaction, which has been produced during the interaction, has been test verified in the wet lab conditions also, where the bioinformatic data and the molecular docking studies has also confirmed their performance, where I am not presenting that particular information in this particular case, where aminocaprylic acid, clindamycin, linoyelaidic acid, mucic acid, 5-hydroxymethyl furfural, nonanol, and acetidine. So they have been tested against the 10 different target sites of Fusarium oxysporum cubensi. So they had the maximum binding energy, which resulted in the suppression, where the multiple mode of action was found to be noticed, and it was found to be appreciated in the interaction of these particular molecules, which we will discuss in detail in some other occasion. Plus here, next to that, since this Bacillus velicensis during the ditrophic interaction, which was able to produce more number of molecules, biomolecules of interest, then we were having a curiosity to understand what might be the treasure which might be present in the genome of this particular Bacillus velicensis. So we went for hunting what are the antimicrobial peptides which is found to be associated with this. So the whole genome sequence of Bacillus velicensis VB7, so the genome size is around 3 MB base pair, which has a GC content of 46.79, and it has a 3090 coding sequences and 8 secondary metabolite clusters. So that was found to be present in the genome of this particular Bacillus velicensis VB7, and it has been confirmed to that of the phylogeny, where the Bacillus velicensis is having very close relationship with that of the Bacillus amelicobacillus, and has more than similarity was found to be very close together, and they are found to be very, very related to each other. So in this case, so what are the 8 different secondary metabolite cluster which is found to be associated with the top of this endophyte Bacillus velicensis, which is found to be associated in this particular investigation is this NRPS, non-ribosomal polyketide synthase, was the region 1 which we have identified, where it is coding for surfactant. The second region was coding for polyketide synthase, which is responsible for synthesis of butyrosine and butyrosine A and B. The region 3 was responsible for terpene synthesis, where the region 4, which is found to be non-ribosomal peptide synthesis, where trans-ATP polyketide synthase, which is found to be responsible for the synthesis of pengazine and beta-lactone. The region 5, the trans-ATP PKS, which is found to be responsible for the synthesis of diphyscidine, and the region 6 was found to be synthesizing bacteriocin, which are found to be varying at different nucleotides, or each and every NRPS, antimicrobial peptides, were having different nucleotides in its length, and the region 7 is coding for the molecule that is these other molecules were responsible for the region 7, which has not found to be precisely detected based on the annotation of this particular genome. So here in the case of region 8, the lantipeptide, which is one of our important key molecule, which is found to be associated with the terp and which is responsible for the synthesis of deficient. So these are the 8 different regions which we have identified from the top of the genome of VB7. So why I am focusing this AMP alone? Because these are found to be having broad spectrum of action. So the synthesis is found to be associated with that of surfactant, basilibactin, bacillazine, diffusidine, fengizine, basilein, macrolactin, calimantine, butyrosine, plantathiazolizine. So these are the various antimicrobial peptide which is found to be associated with the gene cluster or with that of the genome of the bacteria Bacillus amelolecafaciens. So the next curiosity that we want to see is, so how best these particular antimicrobial peptides are found to be having an interaction with that of the target site of Fusarium oxysporum cubensi. So the molecular docking studies will definitely give us a roadmap in such a way that we can identify an organism with a broad spectrum of action or a biomolecule with a broad spectrum of action can be identified. So for which we went for identifying these using these molecule where the protein targets that we used for docking in Fusarium oxysporum cubensi was short chain dehydrogenase reductase. So this short chain dehydrogenase reductase we selected because this is associated with the development of various virulent factors for FOC. And it is responsible for aprisorium formation, conidial germination, penetration, and propagation. Suppose if the molecule that we have selected or the protein or the antimicrobial peptide, if it has a capability to bind with this receptor site, or if it is able to bind with the site, then it can alter the function of that particular one. So likewise, we went for guanine nucleotide binding protein, that is G protein, where it is essential for the pathogenicity. Without this, the pathogenicity may not be there. And mitogen activated protein kinase, it is a development, it's a key virulent factor which is associated in the development of the virulent factors as that of short chain dehydrogenase reductase, and it is responsible for aprisorium formation and conidial germination as that of short chain dehydrogenase. Then calcium channel protein, which is associated with pathogenicity and mycotoxin production. Then GTP binding protein RHO1. So this is associated in this cell wall biosynthesis of the fungus and the virulence of the pathogen. In addition, we were interested to identify how best this molecule can bind with secretory in xylem proteins, which is associated with the virulence. So we tested out how it binds with 6.1, 6.6, 6.8, 6.10, and 6.13, where these 6 proteins, they are found to be as playing a vital role as an effector protein and also favors colonization of the plant tissue and disease progression. So because of this, we have selected these 10 different target sites from Fusarium oxysporum cubensi. So these are the Swiss model 3D structures of these proteins. And if you see the molecular interaction score of this particular target sites with the top the selected antibiotics, that is the antimicrobial peptides, namely basilibactin, butyrosine A, diffusidine, mersacerine, and surfactant, the green color indicates the maximum level of binding energy. So the green shades represent the higher binding affinity of the metabolite, whereas the yellow shades represent the medium binding affinity of the metabolite, and the red represent the lower binding affinity. So if we go for the analysis of this, when we compare all these 10 different target sites, the binding energy with the basilibactin is found to be more irrespective of all the binding site. So if it is the case, the basilibactin, if it is having the capability to bind with all these 10 different receptor sites or with the top the binding protein targets, then naturally the breakdown of resistance may not be possible in this case, where it can give a durable suppression for the particular pathogen over here. So that is the reason why the basilibactin, followed by, in the case if you see, diffusidine is also found to be playing a major role, and mersacerine over here there has a -8.8 and -8, and the highest lower that is binding energy is found to be playing a vital role for suppression. Likewise, in the case of the surfactant also. So irrespective of all these things, all the molecule, they have the binding energy, they have the activity for the suppression, but in the case among all, basilibactin was found to be playing a vital role. So here, this is the docking interaction of the short chain dehydrogenase reductase. So this is the particular one which is found to be associated in the development of virulence factor, germination, penetration, propagation. So if we have tested all these 5 different antimicrobial peptide, so the binding affinity, so this is the one where that is the short chain dehydrogenase protein, where it is at the 3D structure. So the binding energy is minus 10.7 kilocalories per molecule, where the hydrogen bond is found to be associating here, where it is binding with that of the alanine, glutamine, threonine, arginine, and valine. So these are the places where it goes and binds here. So because of the binding of this particular hydrogen bonds with that of the short chain dehydrogenase or the reductase, the functionality of this particular one is getting altered. So because of that, virulence development, apicerium formation, penetration, and propagation, subsequent propagation may be affected. The same might be the case with that of the butyrosine A, where the binding affinity is comparatively lesser, where it is minus 10.7, here it is minus 7.2, where the binding energy. So these are the various different amino acids which is getting bind with that of the hydrogen bond over the protein, the SDR over here. The deficit in the binding energy is minus 7.2 kilocalories. When the hydrogen bond arginine, it goes and binds with that of ARG82 over here. In addition to that, the van der Waals interactions and hydrophobic interaction is also found to be associated with these structures where the binding. Next one is the mersacidin, where the binding energy is found to be only 0.2. So mersacidin is not so effective. So though it is only 0.2, so the lesser the binding energy, the efficacy may not be better. So that is not going to be having a better use for further subsequent investigation. And in the case of the surfactant, the binding affinity is found to be minus 8.2 kilocalories. So likewise, the docking interaction for G protein, that is beta subunit, which is essential for the pathogenicity if we take. Here the binding energy for the bacillobacterium is minus 11.6, which is greater than that of the earlier binding site. So earlier one, which is one, whereas the than that of SDR, whereas in the case of the butyrosine A, it is minus 8.3, which is greater than that of SDR. And the binding affinity for deficitine is also minus 8.9, greater than SDR. And the binding energy for mersacidin is minus 2.6, which is comparatively greater than the SDR, which was only 0.2. Whereas in the binding affinity for surfactant is minus 8.2, whereas in the earlier case, also it was 8.2. So what is that I would like to say here is the binding affinity decides the efficacy of this particular molecule. So there, likewise, the docking interaction of the GTP binding protein RHO1, which is associated with that of the cell wall biosynthesis and virulence of the pathogen over here. So the binding affinity, once again, here the bacillobacterium is minus 11.64, where the amino acids serine, asparagine, lysine, asparagine, serine, then alanine and serine are 149 or 270. So these are the different sites where it goes and binds, and the hydrogen bond is found to be one which is found to be associated with this. And this in turn, as I told earlier, this will result in altering the configuration of the binding site, and thereby the function is getting altered in this particular case. So likewise, in the case of the butyrosine, also the same case where minus 6.7. In the case of deficitine, the binding affinity is minus 8.5. And in the case of mersacidin, it is minus 8.9 kilocalories. And in case of surfactant for G protein, GTP RHO1, it is found to be minus 7.6. So this also has the positive interaction with that of the fusarium target proteins with the docking interaction for calcium channel protein. So this calcium channel protein is essential for the pathogenicity of your fusarium and for the mycotoxin production. So here the bacillobacterium goes and binds with this calcium channel protein in these regions where the binding energy or the binding affinity is minus 9.7 with that of, whereas in the case of the butyrosine A, the binding affinity is minus 7.9. For deficitine, it is minus 9.8. And in the case of the mersacidin, it is 8.8. And in case of surfactant, it is minus 8.6. So here also the binding energy is found to be higher for bacillobacterium where the calcium channel protein function may be altered. Next, in the case of the 6.1, that is secretory in xylem 1, which is an effector protein of the fusarium, which is playing a vital role for colonization and disease progression. the Bacillobacterium has here minus 11.1, the kilocalorie. This is a binding energy or the affinity is found to be more in this particular case, and which is found to be responsible for suppression of the colonization and effector protein action is blocked in this case. So likewise, this is found to be same in the case of the other one also, where comparatively, if you see the 613, if you take, so this is one of the important effector protein, where in the case of the race 4, it is found to be the secretory in xylem 13 is found to be playing a vital role in the case of the as an effector protein, and it also found to be playing a vital role in colonization, and this is progression. So here, to our surprise, the Bacillobacterium here also it had the maximum binding energy of minus 9.4. Beta-rosin has a binding energy of minus 6.8, and defacidine and mersacidine is found to be having a minus 6.4, and surfactant has minus 6.1. So these particular organism, Bacillus valisensis, which is having the capability of producing these different antimicrobial peptides. So when they interact with that of the plant system as a whole, so they have the chances to produce this mixture of antimicrobial peptide, which may be playing a vital role for the suppression of the pathogen during the interaction. So in addition to that, does this endophyte, which is found to be effective through this binding of through molecular docking, so whether they can have the capability to alter the root and shoot architecture of the plant. So this was the next one which was found to be placed in front of us. So we tried the effective organisms like Strenotaphomonas multifila, Brachybacterium paraconglomeratum, Brucella melitensis, Myriades odorotimimus, and Achromobacter xyloxidens through primary and secondary hardening. So these are the seedlings where we went for the primary and secondary hardening. So wherever we go for primary and secondary hardening with the bacterial endophytes, so there the plant architecture was found to be different. The root architecture was different, shoot architecture was also different, the phylochory was also found to be different compared to that of the untreated control, where the plant height and everything was found to be totally altered due to the association of this particular bacterial endophytes, which has been noted in Achromobacter xyloxidens, Strenotaphomonas multifila, then Myriades odorotimimus, next in the case of Brucella melitensis, then Bacillus amelolecufaciens, which is nothing but the Bacillus valisensis, and so on. So we went for documenting what might be the difference in the plant height during primary hardening, secondary hardening, and so on. So if you see in the case, the total cumulative data, so it is around 28.1 centimeter was the plant height. That is the height of the plant was 28.1 centimeter. Whereas in the case of the untreated control, it was 13.97. And the same expression was also found to be noticed in case of Brachybacterium paraconglomeratum, where the plant height was 25.37. So what I would like to say here is, compared to that of the untreated biohardened plants, the plant height or the shoot architecture is found to be altered. And the number of leaf production was also found to be more. It was found to be around 8, that is, it ranged from 5 to 8, whereas in case of untreated control, it was around 5.8, that is, number of leaves was found to be 6, whereas in rest of the bacteria that we have tried, in the biohardened plant, compared to that of Brucella melitensis, all the in other cases, the number of leaves was 2 leaves was found to be more in the case of the treatment, which might be responsible for the increased photosynthetic activity and for the alteration of the root and all the other parameters. So in the case, likewise, in the case of the number of roots and the length of the roots and all these data was also found to be complementing to the finding of the shoot architecture, where the root architecture alteration was also found to be noticed when compared to that of the control. So in the case here, just we went for docking how this photographing, documenting how the root architecture pattern is found to be varying. So this is the case where in the case of the untreated control, where the root proliferation or the root ball formation was not so appreciable level, whereas in rest of the cases, the root proliferation was found to be very better. And here we went for documenting the colonization. So when this found to be noticed, when the architecture, root of architecture and the root biomass is found to be getting increased. So whether this particular organism which you have introduced into this particular system, whether they are found to be sustainable or not, whether they colonize this root system or not. So that was the question. So for which we went for going for the scanning electron microscopic observations where these roots, they were found to be colonized by Bacillus amelolicufation. Here I am focusing only around Bacillus amelolicufation, where all the organisms has been documented. Due to shortage of time, I would like to restrict myself with that of Bacillus amelolicufation within, which is nothing but our whole genome sequence. We confirmed it as Bacillus willisensis VB7, where at 5000 magnification, we can see the complete ramification of the entire root portion by the particular bacteria Bacillus amelolicufation. And here at the case, this is the clear picture where at 10,000 times, if we go for the magnification, we can see the clear-cut bullet-shaped bacterial cells which is found to be colonizing on the root surface. And this is the one where the root hairs which come out of the main taproot system, where are from these root hairs, they were observed under the scanning electron microscopy, where at 10,000 magnification, there was colonization of the root hair colonization was observed. Similarly, this is the photograph which explains at 10,000 magnification how the bacteria is found to be colonizing the root hairs. So likewise, from the root hair, whether the endophyte can move into the pseudostem or not. So there we compare the migration. So whether we took the pseudostem, take the section and observe it, where you go for a sputter coating and then observe it in the scanning electron microscope, where we can see the colonization, complete colonization of the pseudostem also was found to be noticed in the plant system. And this is the one, and besides that, we had a curiosity whether can it move to petiole or not. So the examination of the petiole through scanning electron microscopy also revealed the distribution of Bacillus amelolicufation, the VB7, in the petiole region also. So here, after colonization, so is it having the capability to engineer the plant system to trigger the immune response? So that was the question which was put in front of us to the next one. So the major MAP molecules which are found to be associated with the bacterial endophytes for triggering the resistance response are flagellin, elongation factor, peptidoglycan, lipopolysaccharide, swellanine, cell wall beta-glucan, cell wall chitin, which is found to be responsible for the stimulation of the defense response and growth. So considering that, we were going for identifying some 10 different key genes which is found to be associated with the induction of the defense response in the plant, where we were working on or investigated on chitin elicitor receptor kinase, mitogen activated protein kinase, plant cyclic nucleotide gated channels proteins, then Ricky transcription factor, ethylene responsive element protein, catalase, phenylalanine ammonia lyase, lipoxygenase, then 1-aminocyclopeptide, 1-carbocyclic acid oxidase, and lactase. So these are the 10 different genes which is found to be playing a key role in induction of innate immune response of the plant. So here we went for analyzing through real-time PCR the transcript level of expression, how the different gene transcripts are getting induced. So based on this analysis, if you see here, the Bacillus amelolicufation, this indicates this is the last one, T6 if we take, so that is the healthy control, where there is down regulation is found to be observed in relation to that of Ricky 33 transcription factor. Whereas with respect to the T5, which is the inoculated control, so in the case of the inoculated control, the transcript level is found to be comparatively lesser than that of Bacillus amelolicufation applied one, or in the case of the amelolicufation challenged with that of the Fusarium oxysporum cubensi and Achromobacter xyloxidans, which is T3, and the challenging with that of Bacillus amelolicufation, that is in the presence of Fusarium oxysporum cubensi. So either in the presence of Fusarium or in the absence of Fusarium, the bacteria was able to trigger the immune response, or the transcript of Ricky 33 was found to be induced. So when this Ricky 33 is induced, so this is the cytoscape analysis, how this Ricky 33 gene is found to be linked with that of the other genes associated. So the co-expressed genes associated with that of the Ricky 33 are, this is the, leukoanthocyanidin dioxygenase. So this particular gene is getting, or this protein is getting activated. where this is found to be responsible for the synthesis of anthocyanin, and this anthocyanin is associated with the biotic and abiotic stress management. So this Ricky is linked with this here. In addition to that, if you see the dihydroflavonoid 4 reductase, so this is found to be also linked with the sort of Ricky transcription factor. When dihydroflavonoid 4 reductase is getting activated, the flavonoid compounds is also found to be triggered, which is found to be responsible for induction of resistance response, that is systemic acquired resistance is getting activated. Besides, leucine zipper protein is also found to be induced, or that is found to be linked with that of the Ricky transcription factor, which is found to be responsible for the regulation of the growth hormones. So when this growth hormone is getting regulated, so this is related to that of the abscisic acid, then gibberellic acid, IAA. So all these growth hormones are getting regulated by leucine zipper protein. So when this is getting regulated, probably this may be responsible for growth promotion of the seedling, thus altering the shoot architecture of the banana. So that may be the reason. In addition to that, the WD repeat proteins. So these WD repeat proteins were also found to be induced, and these proteins are found to be playing a vital role in induction of the plant innate immune signaling. So the innate immune signaling is also found to be regulated by the presence of the Ricky factor induction, and besides, all these are found to be transported. So when all these are found to be induced, so the transporting of this from one to the other, so it has to be taken place, which is where ABC transporters are found to be induced in this particular case, and this is found to be also a response in the making the plant to adopt the plant by itself to that of the stress, either to the biotic stress or to the abiotic stress based regulation and adoption is there. So likewise, we went for the assessing the induction of mitogen activated protein kinase in the biohardened banana plants challenged with that of the Fusarium cubensi, and the one without challenge also. As we discussed earlier, in case of inoculated control and healthy control, the level of induction after 72 hours was found to be very, very minimum compared to that of the transcript level of expression of mitogen activated protein kinase, either with Achromobacter xyloxidins challenged or without challenging. Similarly, with respect to Bacillus amyloliquefaciens, the same was found to be noticed. So beyond doubt over here, it indicates that there is that increased level of expression in this particular case. So here what we did, so how, once again, how this particular mitogen activated protein kinase is getting linked with that of the other proteins or other genes associated with it. So this is found to be linked with that of the nitrate reductase. So this nitrate reductase is regulating the synthesis of the ammonia. So this ammonia is utilized as a nitrogen source by the plant and thereby helps the plant for utilizing the nitrogen source. The next one is the PM-APKK, which is nothing but it is found to be associated in signaling cascade for regulating both biotic and abiotic stress are found to be induced by putative mitogen activated protein kinase. Mitogen activated protein kinase. So this is the one which regulates the signal cascade, which is associated with that of the biotic and abiotic stress. Next one, the protein, namely the putative E3 ubiquitin protein ligase KEG, where this is found to be associated with that of the nuclear protein homeostasis, and this in turn regulates the PAMP recognition, and the PAMP mediated recognition is also linked to that of the effector triggered immunity. So this PAMP recognition and effector triggered immunity is getting mediated through the linking of this particular MAPK with that of this putative E3 ubiquitin protein ligase. Next, it also triggers the catalase, so which is found to be playing a vital role in oxidative burst mechanism, which is also contributing to the top the defense mechanism. Next, the receptor type tyrosine. This is another protein which has been induced over here, where this is regulating the protein phosphatase machinery and regulate the mitogen activated protein kinase signaling cascade. So when mitogen activated protein signaling cascade is regulated, all the other events are found to be cyclically linked together, and both biotic and abiotic stress is also found to be regulated. Next, it is also linking with that of serine threonine protein phosphatase, and this is found to be associated with that of the growth and stress related signaling, and this is found to be playing a vital role in auxin transport. So the auxin, which is synthesized by the induction of this MAPK, So this is also may contribute to that of the growth promotion, in addition to that of the nitrogen source present over here, which may be also responsible for the shoot and root architecture alterations and the growth promotion. Next, this is the chorismate mutase, which is one of the interesting part of this particular investigation, where this is the one which is found to be regulating both the cyclic acid pathway. So when it regulates the cyclic acid pathway, this is found to be associated with the other systemic active resistance. Generally, we have learned that the biocontrol agents are found to be associated with the top triggering of induced systemic resistance. But these MAMP molecules from this bacterial origin, they not only regulate the ISR, but also regulate the SIR mechanism, which is found to be test verified using these expressions of the cyclic acid pathway triggering has been noticed by the chorismate mutase synthesis. And the catalase, which the MAPK was found to be also regulating catalase. Likewise, we also analyzed what is the role of catalase in induction of resistance. So here also the same phenomenon has been observed over here compared to that of the inoculated and healthy control, where the oxidative burst is found to be triggered in this case. Hello. So I think
Yes, sir. Audible. You continue, sir.
Okay, sorry, please. Okay, so the co-expression of various genes which has been associated with this catalase, we have analyzed through the cytoscape analysis which has been done over here, where the cytoscape analysis of the catalase, it indicates that where they are found to be linked with that of the RICI44. So this RICI44 transcription factor is associated with the biotic and abiotic stimuli, which contribute for both drought and salinity tolerance. So in addition to the disease reduction, here this is also having a role to suppress salinity. So because the salinity tolerance is also linked to that of the anthocyanin over here, the anthocyanin production, this pigment is also responsible for stress management, where salinity tolerance is also found to be over here, where application of this particular organism or biohardening of this organism of banana biohardening with this particular organism can contribute to that of the synergistic interaction related to that of a stress tolerance related to salinity also. Then the dihydroflavonol 4 reductase, so this is associated with that of the flavonoid biosynthesis. Then WT protein, so this is contributing to that of the innate immune response signaling in the plant. Then this is another interesting phenomenon where aldehyde dehydrogenase is also found to be triggered by the application, by the induction of the catalase over here, where the catalase is found to be responsible for toxic aldehydes over here. So the toxic aldehydes produced in the plant, so either it may be from a different origin. So those aldehydes produced is also found to be eliminated by the induction of aldehyde dehydrogenase. Then tetratricopeptide repeat. So this protein is also found to be regulated, where this is found to be playing a vital role for regulation of the hormones associated with the growth promotion and stress tolerance. And another interesting one that we could observe here is the quinone oxidoreductase. So the promotion or the linkage of the quinone oxidoreductase by catalase, it protects the bacteria from the oxidative burst. So though the oxidative burst is found to be regulated, so that may have an direct effect on the endophytes which is multiplying there, but this particular quinone oxidoreductase helps the bacteria to protect itself from that of the oxidative burst. So that was another one, how this bacteria protect itself in the plant system. So that was the one which we could be able to interpret from this cytoscape analysis. And the leucine zipper protein over here, this regulates the growth hormone, and mitogen activated protein kinase is also getting regulated. So the phospho glucolactonase is another one which is getting regulated, which is associated with another pentose phosphate pathway regulation. So this pentose phosphate pathway is also very, very essential for regulation of the carbon molecules, and the carbon budgeting of the plant is also decided.
Okay, sir.
Please, participant, please mute yourself. Except Nakkeeran sir, please unmute, sir. Yes, sir.
So because I had some interruptions, so just sorry for that. Yes. So the next one, we were interacting of a SERC1, which is a chitin elicitor receptor kinase, where this particular chitin elicitor receptor kinase is associated with the perception and transduction of the chitin elicitor signal molecule. and the fungal MAM biomolecules are also found. So this is the one which is linked here. So here also the same type of observation we have noticed over here. And in this case, the co-expression studies with the cytoscape analysis, which indicated that this circ is found to be associated with that of the protein kinase domain over here. So the protein kinase domain, it regulates the signal transduction pathway. Then protein phosphatase 2C and nucleotide binding kinase is also regulated here. So when protein phosphatase 2C and nucleotide binding kinase is regulated, so this is associated with that of the stress signaling. Then the stress signaling, it is associating, it is linked to it also with that of mitogen activated protein kinase pathway. When mitogen activated protein kinase pathway is linked, the PTI, PAM triggered immunity and effector triggered immunity is also found to be linked in this case. So like we
Malka?
Yes, I think network
I
issue.
think
Network issue, I think. Yeah. We will wait for.
okay.
We will wait. Otherwise, we can contact him.
I will
No, it's
contact. I will
Yeah,
contact.
yeah. I think network issue.
Suddenly power cut off, I think.
Again, him. Joined, sir, all joined. Unmute, sir, unmute.
No, let him join.
Join.
for this because here the power shutdown has.
Yes, suddenly it cut off.
But still, slides are not visible.
Yeah, I think now my voice is audible because there's some shutdown is there. So because of that, I could not get connected. So the another important thing that I would like to stress here is the brassinosteroid insensitive induced. That is brassinosteroid insensitive protein. So this is also getting regulated over here, which is found to be responsible for inducing the signal cascade associated with that of the different genes, cell growth, root growth, and stress tolerance. So in addition to that, the perception and transduction of the chitin elicitor signal and fungal MAP molecules are also found to be regulated by chitin elicitor binding protein over here. So likewise, the lipoxygenase is also found to be regulated over here. And this lipoxygenase, we all know very well that lipoxygenase is found to be associated with that of the synthesis of linolenic acid. That is converting linolenic acid to that of methyl jasmonate and jasmonic acid synthesis there. And this is found to be linked with that of lipoxygenase linked with oxidative, that is catalase enzyme, which is associated with the oxidative burst. And Nakkeeran farm amidase is also found with this particular gene. It's a putative one which is getting induced over here, and it eliminates the toxic metabolites in this particular case. So this is playing another vital role over here for eliminating the toxic metabolites that can be produced either by the plant or the pathogen. And Casperine membrane protein is also found to be regulated, and this is found to be associated in cell wall thickening mediated through that of the lignin biosynthesis. So the lignin biosynthetic pathway is also getting regulated by lipoxygenase and responsive dehydration protein is also found to be activated, which activate the drought stress and regulation of the signal cascade through abscisic acid. In addition, the salicylic acid synthesis is also found to be noticed over here. Next, the lectin is also found to be synthesized over here, where the lectin, we tested for the lectin. So to our surprise, there was an upregulation of the transcripts of the lectin in the case of the biohorn plants with it of this particular bacteria, where the regulation of the lectin was noticed. And this lectin, as we told earlier, all mechanisms associated with these things are found to be getting upregulated, where we Casperine, Nakkeeran formamidase, and protein phosphatase, which is found to be associated with the regulation of PTI initiate. So all these are found to be interlinked with each other, which might be responsible for inducing the resistance response in the plant. So the PAL, which is found to be playing on a phenyl propanide pathway, this PAL is also found to be regulated, where these are the various genes which has been regulated through this particular induction of this PAL gene over here. So this is a nutshell where we are giving an interlinking of various protein-protein interaction. Now, how does all these 10 different genes, they are found to be associated with all these different types of the proteins. So these different types of proteins, so they are involved in regulation of the transcription factor, regulation of the immune response, regulation of PTI, regulation of ETI, hormonal biosynthesis, stress tolerance. So that is the advantage of this particular tool that we are using, so that we can go for a robust tool for identifying a bacteria with a long-standing action, so that it can perform better in the field condition. So this is the enrichment analysis that we have done using the gene ontology based on molecular function, biological process, and cellular functions. So these are the various aromatic amino acid biosynthesis is found to be noticed, and the chorismate mutase, isomerase, then biosynthesis of amino acid, then chorismate mutase eukaryotic type, then chorismate mutase 8 or Q superfamily in eukaryote is also found to be induced over here, and phenylalanine tyrosine is also found to be linked. So these are the various enzymes and the pathways which is found to be linked over here, where this has been grouped into 5 different clusters. Cluster 1, cluster 2, 3, and 4, and 5, where superdomain lysine, that is zinc finger protein, is also found to be linked in this case, where this zinc finger protein is one of the predominant domain which is deciding the responsibility for the resistance response in the plant system. So likewise, the polyketide synthase. So this polyketide synthase through type 3 secretory system. So this is also found to be regulated, where the polyketide synthase is found to be responsible for the synthesis of antimicrobial peptides, which we have seen through that of the genome annotation of Bacillus zoonoses. So likewise, they are also found to be regulated over here. Then thiolase-like activity, then chalcone stilbene synthase is also found to be regulated, and acyl transferase and chalcone synthase. So this chalcone synthase, isomerase, and they are found to be linked to that of the synthesis of salicylic acid, which is found to be interlinked with that of the SIR pathway. So this is the pathway information of the different genes that I would like to highlight over here. So this is the bacterial flagellin, which comes and binds with the plant system on the cell membrane, where the protein FLG22, it binds with the receptor site FLS2, and this FLS2, and it also binds with the this FLS2, it goes and triggers in the membrane or inside the plant system.
Again, I think
Again, he lost the network.
network issue. I think. Yes, again, sir has joined.
Lost from.
Again, joined.
He joined?
Yeah. Sir, kindly share the slides.
Yeah. Sorry, because just now I connected through the cell phone. Now current has come, so that has been disconnected. That is the problem. Okay, so this is the in nutshell that I would like to highlight over here. So when the bacteria, when you go for biohardening, so when it interacts with that of this bacterial flagellin FLG22, when it interacts with that of FLS2, and it triggers the FLS2 goes for in the cytoplasm, it goes and triggers here over here, and mitogen protein kinase is getting activated. So this mitogen activated protein kinase, it goes and regulates Ricci 33 over here. So this transcription factor over regulation over here, and it also this in turn induce the PR1 protein is getting activated over here, and this in turn triggers the PAM triggered immune response. Not only that, the ELF was, that is the EFTU receptor, this EFR is over here. So this goes and binds with this one. So this also goes and regulates this one. And here this FLS2, it also activates the reactive oxygen species also. The oxidative burst, nitric oxide, all those things results with it's a hypersensitive response, and cell wall mediated reinforcement is taking place, which may be responsible for induction of resistance response in the plant system. If we take the bacterial secretory machinery over here, this secretory system, if this goes and triggers the effector triggered immune response by triggering the PR1, where EDS1 is also getting activated over here, where this EDS1 is found to be responsible for induction of hypersensitive response over here, which is found to be responsible for induced in the systemic acquired resistance is also getting activated. In addition to that, alpha linolenic acid metabolism. So when it gains entry into the system, it triggers the alpha linolenic acid metabolism is activated, results in the synthesis of jasminic acid through jasminic acid responsive gene, where MIG2 is getting activated, and ubiquitin mediated proteolysis is also getting activated, where the resistance response can be triggered in this particular case by these proteins. And this MIG2 will regulate all these different types of the different genes over here. So the one is getting interlinked with the various other genes over here. Then phenylalanine metabolism, it goes and regulates the salicylic acid. So this goes and triggers the NPR1, then transcription factor TGA, then followed by PR1 is getting activated, and ultimately will contribute to the resistance response. So after completing the presentation related to that of the global pathogen FOC, now let me move on to that of the next global important pathogen, namely the Phytophthora infestant. So here also, so I'd like to go for the immune response slides along over here. So here we have tested for 10 different genes like RICI33, MIG, lipoxygenase, peroxidase, phenylalanine ammonialase, polyphenol oxidase, catalase, MAPK, EDS1, and PR1. So here also, as I told earlier, in this case, when we go for challenge inoculation, there is upregulation, or the transcript level is found to be upregulated. In case of Bacillus supplensis, which is an endophyte of Phytophthora, that is endophyte of potato, which has been used for treating the potato tubers, and foliar application has been done. After that, the different gene expression has been assessed over here. So these Bacillus amelocupations, along with the Phytophthora infestans, Bacillus supplensis, Bacillus subtilis subspecies speciesini, which when it is challenged with the Phytophthora infestans, they were able to upregulate the transcript level expression of the RICI gene compared to that of the inoculated control and inoculated control. So this is the cytoscape analysis of this RICI33, which I would like to say here. This RICI33, so this is one which is associated with that of DNA binding transcription factor. This is associated with the sequence specific DNA binding and transcription regulation is done by RICI33 through mitogen activated protein kinase signaling pathway and plant pathogen interaction. So these are the one where it regulates extension like protein, putative proline rich protein, RICI, then MAC perforin domain containing protein, and uncharacterized proteins. So this extension like protein is associated with the cell wall assembly and cell growth, whereas this one is found to be the next one, putative proline, is found to be associated with the cell elongation, abscission, and senescence. The next one, which is found to be the MAC, is found to be responsible for MAC membrane attack complex and innate immune response is found to be triggered. In addition, this particular MAC induction is also found to be responsible for lysing the bacteria and kills the virus infected cells also. There is an additional role played by this particular MAC induction. And next is the MIG transcription factor, which we have over here. We have tried where here also, if you see, through real-time PCR, the transcript level of expression, as I told earlier, when you go for challenge inoculation with the pathogen, or even when you use the bacteria endophyte alone in the plant system, it was able to upregulate the transcript expression of MIG in respective of Bacillus supplensis, amelocupations, and Bacillus subtilis subspecies speciesini. And this particular MIG factor is found to be associated with that of the DNA binding transcription factor. It is associated with that of the defense response and positive regulation of transcription is done using MIC transcription factor, and it is a DNA templated one. And this is involving through MAPK signaling pathway. And this is also associated with a lot of jasmonic acid mediated activation of the plant defense. So what is the consequence of this MIC regulation is it regulates the cell function, including proliferation of the bacteria, gene expression of the plant, differentiation in the plant, mitosis, cell survival, and apoptosis. So these are the things which is found to be regulated. This MIC is found to be linked with the pentatrichopeptide repeat superfamily protein, salt responsive protein, mitogen activated protein kinase. It regulates that is JA3, jasmonic acid 3, regulating PTI, ETA, SAR, and ISR response. Then jasmonate and Zim domain protein. Then all these are found to be regulated by the application of the MAC, which is found to be responsible for the resistance response. In addition to that, the PR1 gene transcript level was also found to be increased by challenge inoculation of this particular one. So by this cytoscape analysis, which beyond doubt it indicate that this PR1, it is found to be associated, or it is found to be regulating ATP binding. It regulates enzyme regulation activity. It regulates nitrogen utilization. Where the peroxidase, it is found to be associated with the phenylpropanide biosynthesis, and biosynthesis of the secondary metabolites is also found to be there. The heat shock protein 90 is also found to be regulated by PR1, which is associating the molecular chaperone supporting the correct folding of the newly synthesized proteins. In addition, the TB-like F-box protein and TB-like protein 7 is also found to be regulated, which is involved in protein ubiquitination. So this is one of the essential one which is used for defense mechanism also, which can cleave the unwanted materials. And nucleotide binding ARC protein, this is also getting regulated over here, which is responsible for pathogen recognition and subsequent activation of innate immune response. Next, the RAB is also getting activated, where it is found to be endosome organization. It is associated with cytokinesis and post-Golgi traffic to the plasma membrane and vacuoles. And lipoxygenase, as in the case of the banana, here also we have tested, and this is the role of this lipoxygenase, which is found to be regulating aminocyclopropane 1-carbocyclate oxidase homologs, which is found to be responsible for the ethylene synthesis, which regulates the other hormones associated with it and regulates the plant development process and stress-related responses. They phospholipase A2 family protein, which was also found to be associated in FOC also, where this is an enzyme which cleaves the phospho and galactolipids to generate free fatty acids and lysolipids that functions in plant hormone signaling. So it also, so this is how the one it is getting linked, and the PAL gene is also found to be regulated, where these PAL genes is found to be associated with that of acyl-CoA synthetase, flavonone 3 beta hydroxylone, chalcone synthase isomerase, then 4-coumarate-CoA ligase, dihydroflavonol 4 reductase involved in flavonoid synthesis, chalcone synthase, then trans-cinnamate 4 monoxygenase, which belongs to cytochrome P450 family, responsible for phenylaniline or phenylpropanide pathway, is getting regulated by this one. So ultimately, I'd like to conclude in relation to that of the potato, where the protein-protein interaction network of all these different genes, where there are 10 different genes. So these are the various different genes which are getting interrelated over here. So all these more than 120 genes are found to be linked with that of these 10 different genes, which is going coordinating with that of all the other genes, which may result in induction of resistance response, biotic stress tolerance, abiotic stress tolerance, which can act as biostimulants in growth promotion and all the other things. So these things has to be approved for confirmation in the wet lab. So this is the one where the functional enrichment analysis of the various different genes that we have done over here, where Ricky 33, MIC, LOX, PO, PPO, PAL, PR1, EDS1, MPK, and CAT. So here, if you see the functional enrichment, so we can really appreciate. So all these structures, the round structures, so these are indicating the metabolisms through which various products are found to be synthesized. So all these are found to be getting coordinated over here. And because of this coordination, there is an PAL-PO catalase, peroxidase, polyphenol oxidase, lipoxygenase, catalase. So number of metabolic processes are getting stimulated or initiated at the same time, where several one is found to be complementing with each other for the synthesis of same type of enzymes. So the ultimate pool of these defense molecules may contribute for that of the resistance response in that of the plant system. So this is where this we have to explain in detail, but I think due to lack of time, let me move on to the next one. So this is the one where MAPK signaling pathway is getting activated. And this is the magnesium oriented one and mitogen activated protein, then protein kinase binding, then serine threonine protein kinase, then protein kinase like domain superfamily is also found to be getting regulated in this particular case, where they are also found to be associated with the top various products which is found to be synthesized. So in relation to that, these two different global pathogens of interest, let me also touch upon the work which we have done earlier related to the bacterial endophytes against plant viral disease, where we have published this paper during 2018, where the application of this particular bacterial endophytes, Bacillus valisensis, results in the suppression of tobacco streak virus in cotton, where it alters the root architecture and the shoot architecture also, which is associated with the plant growth promotion, increase of number of flowers, bolls, seed, cotton yield, and reduce the disease intensity. And why it happened? So this we have analyzed, where the application of this, so it has regulated. So all this real-time PCR work, we have confirmed that, and it has been published using this. In the case of microbial pathogens, this particular paper has appeared, where mitogen activated protein kinase, RICI33, NPR1, then PR1, PAL, PPO, LAX, JR, MIG2, and PDF 1.2, they are getting activated over here. So what that we would like to derive a conclusion based on the tritrophic interaction with the groundnut bud necrosis along with that of Bacillus valisensis VB7S. So it is getting linked with that of these genes, where one set of gene is found to be associating for SIR pathway, and other genes are found to be associated with the jasminic acid pathway, and they are found to be interlinked with each other, and this interlinking results in the induction of NPR1 gene through the activation of the signaling cascade events mediated through that of the transcription factor which takes place in the nucleus. So the nucleoplasm, where the regulation of the defense response, flagellin, elongation factor, tubulin, so they recognize that it induced this mitogen activated protein. So this is the graphical abstract that we would like to present based on the induction of the immune response induced in the plant through the triggering of innate immune response. So finally, I would like to conclude that the whole genome sequencing will really helpful to understand what is the treasure which is found to be present in a particular bacteria, where that could be explored better for the management. In addition, that is a team effort with that of the bioinformatic persons, where the molecular docking and cytoscape analysis, molecular simulation will help in unraveling the mechanism and action of the biomolecules, by which we can go for picking out of a robust biomolecules which can be explored for the management of the plant pathogens in whole. And thus the map molecules from different endophytes can be selected, and that can be exploited well for giving a sustainable solution for the long-standing performance under the field condition, where though we publish in a high impact factor, it has to be translated into the product, for which this type of basic work is very, very essential. And engineering of the endosphere with the microbiome will really open new avenues, and it can open the roadmap for coming out with a lot of new solutions. And I once again thank for the society, where I am also one of the life member of IPS, where I thank Dr. Pratibha Sharma Madam, who was really giving this particular opportunity, and Dr. Robin Gogai, and Dr. The present President Pandesji, and all the other members and the fraternity of this society for giving me this opportunity. So this is where I am only representing the work done by this particular team of group of student community, where this is the one where I am the messenger for these people. It is the work done by them under the support of the grant given by us. So it was made possible only with this team. Without this team, it is not possible. to present in front of you and to stand in front of you to deliberate this particular lecture. So let us live and grow with endophytes for better prosperity, for increasing the income of the farming community. Thank you for the opportunity, please.
Thank you, sir, for your wonderful lecture and very knowledgeable lecture on understanding the endophytome mediated induction of local significance. Now, one or two questions are invited from the participants.
I don't have question. I have appreciation to Professor Nakkeeran that how deeply he has elaborated. And Dr. Gagoi, if you are here, Dr. Gagoi, Robin.
Sir, they have
Hello,
lost the
Robin
connection.
Gagoi.
I think electricity
Robin
is not there.
Okay, in future, at least we will invite him for the review in Indian phytopathology, because such a deep study will be highly useful not to only few people, for the audience of crop protection. Such a wonderful lecture for the first time I am hearing. Really great lecture. Congress, Dr. Nakkeeran. Very good. And your student also.
Thank you, sir. Thank you. Thank you.
I'm audible, sir. Can you hear me?
Yes, 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 sir. Yes, sir.
Sir, myself, Dr. Satish Verma, I'm working in Department of Botany, Banaras Hindu University.
Okay.
And I also works on bacterial endophytes, especially the seed endophytes. So my question is that, sir, because you have inoculated the bacterial endophytes on the tissue culture plants, means generated from the tissue cultures. So,
Okay.
at what time you have inoculated? It was up during the hardening process or before the hardening process?
So this is where we had to, biohardening has to be done through primary, at the time of primary hardening, and at the secondary hardening, where we have given one application during primary hardening and during the secondary hardening. So after that, it will be challenged with that of the pathogen. So this will help where we go for regulation of the studies of this particular different gene expression were found to be observed there. So the application has to be done at the time of primary and secondary hardening. So that because why I'm saying this is, when you do at the stage of primary and secondary hardening, because the seedlings are going to be given to the farmers. So when you develop a seedling with resistance response, then that can help in immune response in the plant, so that it can counter the infection of the pathogen when it is planted in the field condition.
Okay, sir. One more thing, in your bioinformatics work, so you have shown that the this bacterial antifungal peptides like surfactants and the others, they interact with the G proteins of the fungi, and might be the reason behind the suppression of the pathogens. So what do you think about this surfactant may interact with the G proteins of the host also, the plant? So what is your view on that? Because the fungi G proteins and then the host G proteins both have a receptors, these G proteins. So is there any your finding or docking experiment finding regarding that, the interactions of this?
Okay, this is a very good question. So how best the G protein coupled receptor is found to be interacting with it of both the plant, that is both the plant, and with it of the bacterial in this case. So what is the case here is, so this is one of the one where we have done the work related to that of the bacteria alone. So I think this will open another researchable issue for our students, so they can try to interact how this is going to react with it of the G protein. Probably, I hope so. There may be minor variations in the G proteins in the plant origin. So whether that type of recognition is there or not, so that we have to think, and then we have to rework on that particular issues. I think that will also be another researchable issue which we can think of.
Okay, sir. Thank you very much, sir. It's a lot of appreciation. You did a lot of work, and it is a motivation for us, the younger means I'm working in the Endophytes, so it is a great motivation for me. Thank you very much, sir.
Thank you, sir. Thank you.
Dr. Nakkeeran,
Sir, how are you, sir?
fine. So I had both the now thunder showers, outside Coimbatore also and your lecture also. Congratulations for a very good presentation. I think young students and scholars would have been benefited by that. So now, what about the endophytes? These bacillus amelolicupaceans are equally effective bioagents. That registration process, how for endophyte anything is, can it go along as a plant growth promoting organism like that, or as a bio?
Sir, suppose if you are claiming, I think you also know that if you are claiming it as a biopesticide, naturally we have to go for thing. But if you go for biostimulant, I think we can proceed, or even as a, since it is found to be associated with abiotic stress management and with it of the regulation of the growth hormone and all the other things, as microbiologists claim, as a biofertilizer angle also we can think of it. We can go for immediate promotion where we can claim as a biofertilizer, so that which can do the dual purpose. In that angle also we can take it immediately to the top of the farming sector.
So like that, we have so many centers, they have very good strain, potential strain, but they are unable to take it forward. So if it is other way, if it can be promoted as a biostimulant, anyway, it is going to be a systemic thing. It is entering almost going till the leaf sheath, So does it go to fruits also?
No, we have not checked up to fruit, sir. We have checked up to the petiole we have seen. Yeah. And in the leaf also, but above that in the fruit, I think already there is a pre-existing microflora where Bacillus subtilis and all the other are found to be present in the nectar and in the case of the fruit also.
Definitely,
But what is the thing is we are unable, what is the demerit in this particular case is we are unable to develop a tracking system of the introduced bacteria. Okay. So just we can compare between the untreated control and the treated control, and that is only because the labeling is found to be very tough with the gram-positive bacteria. Yeah. So if we do that, then I think we can trace even up to fruits also.
Okay. Again, thank you for your very good presentation. I'm sure IPS really they have done a wonderful job by inviting you for this lecture. Thank you.
Thank you, sir. Thank you, sir.
Yes, Dr. Nakkeeran.
Yes, please.
Congratulations. I'm very happy that you have given an excellent this presentation covering these three major pathogens. So Bacillus amelolicupaceans. So did you characterize the VOCs?
Yeah, we have done. We have characterized volatile organic compounds also. So that we are having a separate presentation. I think that will have. I think we have also published that paper also. Okay, in those things where we recently published that one in in environmental microbiology, where what are the VOC which is found to be induced during monotrophic interaction, ditrophic interaction. So the supplement it is present in the supplementary file of the journal. Around 30 pages supplementary file is there. Suppose if you want, I can send the link. Number of informations we have floated in that.
Okay, thank you.
So, sir, I have one query. Nice presentation, sir.
Thank you, sir.
Whether you identified the virulence genes in the pathogen or either during interactions?
No, because we were not in the process of identifying the virulence gene, because our motto was management, all those things. So that particular part of work is carried out by several other group of people. So you don't like to enter into that particular area. Ultimately, we would like to address how to manage the issue, for which we want to have a scientific background information. So for which we went for exploring these endophytes.
So my question is, sir, whether genomics is better approach or proteomics is better approach to identify some genes related to during interactions?
During interaction, I think it is better nowadays. The genomic perspective also very clearly explains what is the endpoint of the gene results in what is the protein. So that can also be annotated very well if you're having a very good bioinformatics with you. So based on that, genomic can be a robust tool by which we can easily identify that also.
Because nowadays transcriptomics are also coming in new in molecular.
Transcriptomics also a very good tool where we have also done the transcriptome analysis, which I could not present in this because time is a restriction for us. We done both for potato and for banana also. We have done the transcriptome analysis, where the money is one of the major issue in this particular case. If we have money, anyone can do that work, because our part, without money, research is not possible.
Yes.
So that is the where most of them are unable to, though they may have a better research IQ and research thirst, the lack of finance and fund is the stumbling block for these things.
Okay.
So yeah, Dr. Malkan, there are I think many questions we could see in the chat box. Whether 2-3 questions you can read out?
I can see, but sir, during network I didn't see the questions in this chat box.
Yeah,
Dr. Parmeshwari can read the questions earlier. Dr. Thangavilu sir also asked the questions.
So here, what is the thing is, Dr. Thangavilu has raised a question.
Yes.
YKM5 and Karpuravalli both are susceptible to both race 1 and TR4 of FOC. So this is the question that he has posted. So regarding, based on the literature collection that we have collected, YKM5 has resistance to the information that is against Fusarium wilt. Probably as he is having a continuous association in banana, so probably he might have found out that it is found to be susceptible, which we will be rethinking on that, how best this can be worked out. And the YKM5 is resistant to nematode and not to Fusarium wilt. So that was the thing that he has given. But in our case, that based on the literature collection, I was unable to find the YKM5 is found to be susceptible to Fusarium wilt. So if he is having the information, the susceptibility, the published information, I think that will be better for us for doing still more a better research in this particular line. So I request Dr. Thangavilu to give me the literatures associated with these issues, so that will help us to fine tune our further research program. And next, Dr. Ritu Mawar, she has given, that is only congratulations given. I have an urgent call. Then Sahil Mehta, how was the enrichment picture was generated with the 10 protein together at the same time? So here we are having a specific software for pulling all together. So that is an exclusive software in our bioinformatics department we have. So based on the help of the bioinformatic people, we have done those things. I think if you want this specific software program, we can inform what is that particular program over there. And Dr. Anil Kumar, so informative, very nice presentation, very good lecture. Thank you, sir, for nice lecture. IPS should plan for workshop on endophytes and plant defense, and Dr. Nakkeeran should deliver the lecture. That is someone you have given like that, Dr. Muhammad Suhaib. And I think
last question, sir. The endophytes of medicinal plants should prove more effective against plant pathogens.
So I think there was another lecture which I gave in another platform where they have identified endophyte from that of a medicinal plant, and they have transferred that particular endophyte to suppress Pseudomonas solanacearum in another plant, where this was able to suppress that particular pathogen also. But what is the issue over here is the adaptability of a bacterial endophyte from a medicinal plant to another plant. So it's a sustainability, multiplication, and all those things has to be test verified. If it is working well in that particular case, there is no harm in exploring those endophytes also. So for that only I'm saying endophyte is having a really, it will have a very broad opening roadmap has been opened. So everyone can work for even another paper that we are going to, it is in progress with the editor, where the Stenotrophomonas multifila. So that is another endophyte, which is also playing a very, very important role, which will be getting published in a short time. So likewise, we have sequenced more than 20 different genera of bacterial endophytes, which gives suppression of the plant pathogens. So opportunities are very wide. It depends how we tap those things.
Thank you, sir. Now I request Dr. Malkan Singh, Treasurer, IPS, to provide vote of thanks.
Good evening to all. On the behalf of society, it's my great privilege to propose vote of thanks in the celebration of Platinum Jubilee of IPS, Indian Phytopharmaceutical Society. Society is celebrating 75 years of journey. Dr. S. Nakkeeran, Professor, Department of Plant Pathology, TNU, who spent his busy time to deliver this talk, understanding the endophytome mediated induction of defense genes for management of pathogen of global significance. He mainly emphasized the diversity of microbes, culturable endophytes, bacterial endophytes, and especially Bacillus thuringiensis, and also FOC, bacterial wilt of fungal wilt of banana, and genome sequencing of bacteria and annotation part of these genomes, and secondary metabolites and docking of proteins and interactions, and also the induction of resistance due to these bacteria, and also late resistance in the late blight of potato, and apart from the defense genes which are upregulated and downregulated during the application of the bacteria biocontrol agents. Apart from that, our president, Dr. Pratibha Sharma, today she couldn't join because of network. She's always made a sincere effort to organizing, coordinating, guiding all the committee members, and initiated this program in the occasion of 75 years of the society. And our president-elect, Dr. Rakesh Pandey, who always behind that suggestion to conduct this Platinum Jubilee celebrations. And Dr. Robin Gogoi, Secretary, IPS, he always behind us all the lecture series of IPS, and all organizing committee, particularly this Platinum Jubilee celebration event. Dr. Prashant Jambolkar, Dr. Parameshwari, Dr. Pramod Kumar Gupta, Dr. Sushil Sharma, Dr. Sangeeta CG, and Dr. Amlendu and Dr. G. Prakash. And all, even though in Zonal President Dr. Riti Mawar today is given the welcome address, and all the senior executive council members of the society, all Zonal President of the society, and senior editors and editors of the society, and I would like to thank all the IPS members today who joined this program. And finally, I would like to thank all IPS staff, and thanks to one and all. Thank you very much.
So we will be meeting again coming Wednesday.
Yes, sir. Yes, sir.
Yeah.
Thank you.